Glycolysis is also known as Ebden-Meyerhof-Parnas pathway. This is the name of the scientist who identified this pathway, the three scientists' name. So the food, as I told earlier, it is from the food is what we get the energy. So how the food is processed so that we get the energy. One method or one pathway is the glycolysis, wherein it processes the glucose molecule so that ultimately we get energy. We can define glycolysis as an enzymatic pathway, okay, because it carries series of enzyme reaction wherein the glucose is 6 carbon molecule. It is converted into 3 carbon pyruvate molecule with the formation of ATP and NADH. So from the glucose molecule, by series of enzymatic reaction, the end product we get 2 molecules of pyruvate and ATP and NADH. So these are the energy rich currencies. So where does this glycolysis occur? The glycolysis occurs in all the cells, that is the cytoplasm. It can happen under the aerobic or under the anaerobic conditions. Particularly, it is important for the matured RBCs because we know that the RBCs lack mitochondria, and even when we are doing vigorous exercise wherein there is low level of oxygen, there also it becomes an important process. So we can divide the entire flowchart of glycolysis into two phases. One is the first phase is the preparatory phase, and the second phase is the payoff phase. So the first phase, it is called preparatory phase. Herein, two ATPs are used up. Okay, they are utilized during the reaction. Whereas in the second half of our cycle, it is known as a payoff phase because here the ATP is generated. So also 2 molecules of NADH is produced. So if you look into the glycolysis, the first what we are going to speak, that is this preparatory phase, that is the energy consuming phase. Okay, the energy is utilized here. So energy means immediately what should come to your mind is the ATP. Here in preparatory phase or the energy consuming phase, we are utilizing, our chemical reaction is utilizing the ATP. So our starting molecule is what? Glucose. It is a carbohydrate. It is a 6 carbon molecule. This glucose, it gets into our small intestine and then into the bloodstream. So now your blood sugar increases. Why? Because there is glucose from your. So whatever the glucose is present in your body from your small intestine, okay, it enters into your bloodstream. Okay, it enters into your bloodstream. Here what is happening? So there is an increase in the sugar level or the amount of glucose in your blood. So immediately what happens is your pancreatic cell starts producing insulin. So the glucose utilizes a glucose transporter wherein it's present in the blood, that it utilizes a glucose transporter. Once the glucose is inside your cell, it cannot diffuse out. Okay, if this is your cell and if the glucose is present here, okay, it cannot again diffuse back, because once inside the cell, this glucose will be processed by the enzyme kinase. How? That is, this enzyme, the kinase enzyme, it starts adding a phosphate group to the glucose molecule. It starts adding a phosphate group. Here you can see. Okay. From where did it add the phosphate group? It added the phosphate group from the ATP. So your ATP gets reduced to ADP. So now what happens? You get the product glucose 6 phosphate. So this glucose 6 phosphate that has a configuration which is different from glucose, so it cannot escape from the cell. So it is like trapped inside the cell. So since an phosphate molecule is added to the glucose, this process is known as phosphorylation. So now what has happened? Your ATP is utilized. That is what we told, it is an energy consuming phase. So you have lost one ATP molecule. So here you can see the enzyme which is used is the hexokinase. There is also another enzyme known as glucokinase. Okay, both have the same function. What is the difference is the hexokinase is present in the cells. Okay, this is hexokinase is present in the cells, whereas your glucokinase, okay, it is present in the liver. That's the difference. It is present in the liver. So glucose 6 phosphate means, that is, your phosphate is added to the sixth carbon. Now, if, for example, this is carbon 1, 2, 3, 4, 5, 6. Okay, so here it will be added, your phosphate group. So that is why it is known as glucose 6 phosphate. So in the next step, which is, you can see here, the glucose 6 phosphate. This in turn is converted into fructose 6 phosphate by the enzyme phosphoglucoisomerase. So this process, okay, is known as isomerization. And here you can see that the arrow mark what we have put is irreversible. Remember this also, it is irreversible. So the enzyme what we have used is the phosphoglucoisomerase. So we got the product fructose 6 phosphate. It is same. In the sixth position, the phosphate is added. Only the glucose is just converted into the fructose, the isomer form. Then we have the fructose 6 phosphate, which is converted into fructose 1,6 bisphosphate by the enzyme phosphofructokinase. So again, you see the word kinase come into picture. That is addition of a phosphate group. From where? From the ATP. So again, this enzyme phosphofructokinase, it uses the ATP so that the phosphate of one of the ATP goes and binds to this fructose 6 phosphate. At which position? At carbon 1 position. Okay. So it is fructose 1,6 bisphosphate. So again, what has happened? We have lost one more ATP. So together we have lost 2 ATP. This is a rate limiting step. Okay, that is, it determines the speed of the reaction. which all glycolysis happens. So this is an important step. It is a rate limiting step. It determines the speed at which the glycolysis is occurring. In the next step, you see that the fructose 1,6 bisphosphate, it is cleaved by the enzyme aldose into 3-carbon glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So, this dihydroxyacetone phosphate, okay, it is gets converted into glyceraldehyde 3-phosphate. Because only glyceraldehyde 3-phosphate, only when it is present in the form of glyceraldehyde 3-phosphate, only then it can continue the glycolysis. It can continue the pathway, not in this form. So this dihydroxyacetone, by using another enzyme, isomerase, it gets converted into glyceraldehyde 3-phosphate. So how many glyceraldehyde 3-phosphate now I have? I have two. One this and another one this. That is, this is also got converted into glyceraldehyde 3-phosphate. So here we see that we have utilized 2 ATP. That is why it is known as the energy consuming phase. Now, from here you have to remember that the entire cycle goes twice because we have 2 glyceraldehyde 3-phosphate. So, the next step, what happens? Your glyceraldehyde 3-phosphate, okay, is converted into 1,3-bisphosphoglycerate in the presence of the enzyme glyceraldehyde 3-phosphate dehydrogenase. And here what happens? Your NAD is reduced to NADH. So from here the steps occurs twice because already we have told that there are 2 molecules of glyceraldehyde 3-phosphate. So how many NADH is formed? This also 2 NADH is formed. Okay, and this NADH in turn, this enters into your ETC cycle, which we shall deal it later. Okay, ETC cycle. Your NADH, which is formed here, 2 molecules of NADH which is formed, it later on enters into the electron transport chain, wherein it gets converted into 3 molecules of ATP. In detail, we shall deal it when we talk about this particular chapter. So I have ended up in what? 1,3-bisphosphoglycerate. Okay. So I at the position 1 and third carbon, first carbon and the third carbon, I have a phosphate group. That is the meaning of this 1,3-bisphosphoglycerate. In the next step, we can see that the enzyme phosphoglycerate kinase, okay, it removes one of the phosphate group. It has removed one of the phosphate group is removed, okay, and it is gets converted into 3-phosphoglycerate. So this phosphate, whichever is being removed, it binds to the ADP. Okay, so this phosphate which has been removed from here, it adds to your ADP and it gets converted to ATP. So how many ATP are formed? 2 ATP are formed because I told you the step occurs are occurring twice here. So 2 ATP molecules are formed. Don't get confused because since glyceraldehyde 3-phosphate here, it is formed 1 here and this also gets converted into 2. Now twice the steps occurs everywhere. So we have got 2 ATP molecule with a product 3 phosphoglycerate. That is only here the structure. If it is like this 3 phosphoglycerate, only the phosphate group will be present only here. Okay. So in the next step, we have the enzyme mutase. It removes the phosphate on the 3 phosphoglyceride, making it into 2 phosphoglycerate. That is into, where is the phosphate group? It is on the second carbon here. So your mutase, what it does, it converts the 3 phosphoglycerate into 2 phosphoglycerate here. And the next step, we have the enzyme enolase. It removes the water. From where it removes the water? It removes the water molecule from 2 phosphoglycerate. Your enolase, it removes the water molecule here. You can see here, it has removed the water molecule and then it gets converted into phosphoenol pyruvate. What gets converted? Your 2 phosphoglycerate gets converted into phosphoenol pyruvate by the enzyme enolase, wherein it removes a water molecule from this compound, 2 phosphoglycerate. So I have ended up with phosphoenol pyruvate. Finally, the enzyme pyruvate kinase transfers a phosphate from phosphoenol pyruvate to ADP, making pyruvate. This pyruvate is a 3-carbon compound. From here, everything is 3-carbon compound. So how many pyruvates are formed? 2 molecules of pyruvate is formed. So remember again kinase. Whenever kinase comes, remember that there is formation of ATP. So you see here, that is a phosphorylation reaction. Here you can see phosphoenol pyruvate. Okay, the presence of the enzyme pyruvate kinase. It removes the phosphate group. Okay, so it has removed the phosphate group and this phosphate group binds to the ADP and it forms an ATP, forming the byproduct 2 molecules of ATP and 2 molecules of pyruvate. So here the end product, pyruvate is our end product, right? So how many ATPs are formed? So here is 2 molecules. So I have got 4 molecules of ATP. And how many molecules of NADH I got? I got 2 molecules of NADH. NADH. And what else did I get? I got 2 molecules of pyruvate. Okay, so I started up with what? Glucose, which is a 6-carbon molecule, glucose, and I've ended up in 2 pyruvate molecule, which is a 3-carbon compound. So the enzyme pyruvate kinase has transferred the phosphate group from phosphoenol pyruvate to ADP, making pyruvate and 2 molecules of ATP. So this is also an irreversible reaction. Now, here you can see this last step I have given the full explanation. This is your phosphoenol pyruvate, 3 carbon are there. Okay. So here in the second carbon, you can see the phosphate. This phosphate is removed. And when it is removed, what we get is the pyruvate. Now, sometimes what happens is some cells do not have access to sufficient oxygen, like your skeletal muscles, particularly when we are doing strenuous exercise, vigorous exercise, or your RBC. They lack mitochondria. In such situation, the cell uses the enzyme lactate dehydrogenase. Okay, under these conditions, our cells, whenever there is low oxygen, okay, then our cells uses the enzyme lactodehydrogenase and it converts the pyruvate into lactate. Okay, so how it this enzyme lactate dehydrogenase, it utilizes this NADH plus and it gets converted to NAD. Okay, so this step we have to remember. So this usually occurs in our skeletal muscles and RBCs. So one point you have to notice, this NAD already we have come across. Okay, NAD is required to work. It works along with your glyceraldehyde 3-phosphate dehydrogenase, which is required for your glycolysis to keep going on. So this lactate, whatever has been formed, okay, this lactate which has been formed from your body is removed from blood by the kidney. But sometimes what happens is this lactate, it builds up very fast in our body. And that is why you can see sometimes the muscle sorenesses, okay, when you exercise. This is because of the very soon the lactate has been built up in your skeletal muscle. So this is about our glycolysis. In the next part, we can study about the regulation. But before we wind up, let us see the net gain, what we have got. That's earlier what I have mentioned. So I have got 2 molecules of pyruvate, 2 molecules of NADH, okay, and 4 molecules of ATP. But in my previous steps, that is in the preparatory phase or energy consuming phase, what happened? We utilize 2 ATP. Okay, so what happens from here? If you minus 2 ATP, you are left with only 2 ATP. Because 2 ATP was utilized in the first step and 4 ATP molecules are produced in your second phase, 4 minus 2 is you are left with 2 ATP. So this is your net gain in your glycolysis. This is how the glucose from my food molecule is converted into pyruvate, and from this you will see in the next cycle what happens to the pyruvate. Okay. And it forms the, it produces the ATP molecules and NADH molecules. So the question what we can ask is whether the glycolysis is an anabolic pathway or catabolic pathway. Yes, the answer is it is a catabolic pathway because it is breaking down the glucose. Okay, how we define catabolism? It is the breakdown of the glucose molecule. So the glucose is broken down into what? It has broken down into pyruvate. Catabolism is breaking down. And also we defined how catabolism, we told that here energy is produced. Okay, here the energy is produced. Hence, glycolysis is a catabolic pathway. We shall next continue.